This study will demonstrate the findings of Mayer and Bartel's 2009 study titled Widespread Shortening of 3' UTRs by Alternative Cleavage and Polyadenylation Activates Oncogenes in Cancer Cells. When DNA is transcribed into mRNA, it undergoes multiple forms of processing, including polyadenylation. As you can see here, mRNA transcripts have multiple different sites of poly A tails where they can be cleaved. This cleavage in different places due to alternate poly A sites can lead to shorter isoforms as shown and longer isoforms of the mRNA transcripts. The shorter isoform was shown to have increased stability, increased protein expression, and increased oncogenic activation. This was supported by doing an mRNA half-life assay. This assay showed that the shorter isoforms had greater stability and therefore lasted longer and expressed more proteins as opposed to the longo isoforms, which were degraded and did not last as long to produce as much protein. Due to altered cleavage, the shorter mRNA isoforms lack the Let7 miRNA binding sites, which are known to silence or repress expression of the mRNAs. mRNAs silence transcripts by using the RISC complex, or the RNA-induced silencing complex, that splices mRNA transcripts thanks to the mRNA complementary binding. These splice products, due to RISC and mRNA binding, are then degraded. Using the IMP1 gene as a model, Mayer and Bartel produced long and short mRNA isoforms and conducted a soft agar assay over time in order to test for oncogenic transformation of these cells. It was shown that the IMP1 shorter mRNA isoform actually produced more oncogenic transformations over time, as predicted, due to the shortening of their 3' UTRs thanks to alternative polyadenylation and cleavage. Mayer and Bartel were able to conclude that there may be a high incidence of APA in cancer cells with consequent loss of 3' UTR repressive elements, suggesting a role of alternative polyadenylation in oncogene activation without genetic alteration. This provides basis for potential RNA therapeutic targets in future studies.